Hello, everyone. So we're going to talk a little bit more about the molecular basis of evolution. And we're specifically going to talk about different types of mutations today. So we've already talked about mutations, and I want to make sure that we're all on the same page with the definition of mutation. So I've written here that a mutation is any change in DNA sequence, and that includes things that are large or small. Well, whatever change there is from one base pair to another, that counts as a mutation. So this could include single nucleotide polymorphisms, as we see here. They are also referred to as SNPs because that's a lot easier to say. So again, single nucleotide polymorphisms, and those are a change in a single base pair. So we talked about this with respect to the sickle cell gene, and sickle cell is a change in a single base pair or nucleotide. And it's not a frame shift or anything. It's just one base pair changes to another base pair. And this causes the phenotype to change quite dramatically. It causes hemoglobin to not act as it's supposed to and to clump up and form long fibers, which then distends the blood cells into this sickle cell shape. And if you want to learn more about that or review it, then you should take a look at my YouTube channel. I believe that there is a video on that there. And so again, that is caused by a single base pair change from the quote unquote normal sequence to the quote unquote disease sequence or the disease state. Now that's called a mutation. Also referred to as a mutation would be very, very large transfers of genetic data, for example, in horizontal gene transfer. So this is most commonly something that happens in bacteria. And the other day we were talking about antibiotic resistance. And oftentimes when a bacterium acquires antibiotic resistance, it's not because they had a single change that arose naturally or a mistake from DNA polymerase copying the DNA or even environmental damage. Usually, antibiotic resistance in bacteria is, or a single bacterial individual is acquired through horizontal gene transfer. And this is when a large amount of DNA in particularly a plasmid. And so a plasmid is circular DNA that is not necessarily part of a genome, but oftentimes plasmids contain several genes, and sometimes they also have origins of replication so that they can cause whatever cell they're in to replicate them as the cell replicates. And they're mostly found in bacteria. And so a single plasmid can contain many genes or several genes. And these can actually be transferred from one bacterial individual to another all in one transfer event. So bacteria, I'm not going to go into it too much because we're not a microclass, but bacteria can actually send out this special structure called the pilus, and they will send the DNA along this protein structure, and the other bacterium will take that DNA in, and it all depends on how long they're connected, but they could transfer thousands of base pairs this way. And so you could be some bacteria without any antibiotic resistance at time point 0, and then an hour from now, you might be able to be resistant to a couple of different antibiotics because you got that gene transfer. And so this could be a large amount of DNA that's transferred by a plasmid, which can contain several genes. And so both of these, to my point, are referred to as mutations, both the single nucleotide change and the horizontal gene transfer, which can contain many genes. These are both referred to as mutations. So you really have to know what type of mutation you're referring to when you use that word, or you need to figure out what someone else is referring to, because mutation is a very general term. And so in the next video, I'm going to talk a bit about different types of mutations that really are just of this smaller variety in terms of their SNPs, and how even different SNPs are not equal in terms of how they affect the phenotype.